Good afternoon. Welcome to MSK Science Spotlight. I am your host today. I am Danwei Huangfu. I'm an associate member in the developmental biology program here at Sloan Kettering Institute. This seminar series features leaders of today's scientists in basic and translational biomedical research. If you have questions throughout the talk, you can email them to the Office of Scientific Education and Training at OSET, O-S-E-T, at mskcc.org, or by tweeting at MSK Education. Questions will be addressed at the end of the program. Now, let me introduce today's speaker, Dr. Scott Lowe, our cancer biologist. He is the chair of the Cancer Biology and Genetics program here at MSK. He's the chair of Jeffrey Brain Cancer Research Center at Memorial Sloan Kettering Cancer Center. He's also an investigator of the Howard Hughes Medical Institute. Scott Lowe has used the genetic engineered mouse models to do cancer discovery, and also using RNAi approach to discover tumor suppressor genes. To me personally, he has been an inspiring leader in cancer genetics. So I'm very much honored today to host his talk. His talk today is entitled Targeting Cellular Senescence in Cancer and Beyond. Scott.
Thanks, Danwei. It's a great pleasure to be part of this MSK Science Spotlight series. I think I'm the third one so far, and there'll be a few more, so stay tuned. And I'm looking forward to telling you about some recent work from my laboratory. Just before I start, I want to acknowledge my disclosures and then tell you that my lab has been interested for many years now in what I like to call tumor suppressor networks. These are processes that restrict the progression of cancer cells. Many of them are controlled by tumor suppressor genes. These are genes that are inactivated during the course of tumor evolution. And we study these networks because we believe they point to physiological processes that naturally protect the organism against cancer. And we like to understand how these processes work, what they normally do, and how they're altered during tumor development. We ultimately believe that through this understanding, it might become possible to re-engage tumor suppressive networks, and that could have potent anti-cancer effects, leading to a next generation of new therapeutics. Now, I'm going to talk about one of the tumor suppressive programs that my lab has studied now really for almost 2 decades, and that is the process of cellular senescence. And just by way of a little background, senescence was described really 40 or 50 years ago by Hayflick as the property of cultured cells that underwent replicative exhaustion in vitro. These cells would grow, they'd stop dividing, they become insensitive to mitogens, and essentially never divide again. Now, we know that over the last 20 or so years, it's become clear that senescent cells act in sort of a stress response program that can limit the proliferation of oncogene expressing cells. And you could imagine if oncogene expressing cells can't proliferate, they can't form tumors. Nevertheless, this program is disrupted during the course of tumorigenesis, such that most tumors that arise in mouse models or in human patients have evaded this process. Now, senescence probably didn't arise simply to suppress cancer. We believe it has a physiologic role in certain wound healing responsive and regenerative processes as well. And I'll come back to that throughout the course of my talk. Now, there's been a lot of work on understanding the roles and regulation of senescence, and we understand a fair amount about the molecular mechanisms that drive this program. One of the sort of most well-studied and traditional part of the program is the stable cell cycle arrest. We know that senescent cells activate tumor suppressors, such as p53 or the retinoblastoma protein, and that leads us to changes in chromatin structure that ultimately repress essentially the expression of proliferation-associated genes. So this is a gene repression program that is important for the stable cell cycle arrest. But there's another component to the program known as the senescence-associated secretory phenotype. This is a gene expression module that's controlled by NF-kappa B, the transcription factor, and the chromatin reader BRD4, leading to the upregulating of genes that encode factors, most of which are secreted, hence the name SASP, and many of those are known to modulate the tissue environment, some of which can actually promote and modulate the immune system. So again, when cells undergo senescence, these both components are activated, and we believe that contributes to the tumor suppression features of the program. Now, this immune modulatory effect is something my lab has been studying over the last now several years, or a decade or so. And I just want to illustrate sort of how this can look in a simple co-culture assay where we can take senescent cells, in this case labeled in green fluorescent protein, and co-culture them with an immune cell, natural killer cells, which are labeled in red. We've also made the senescent cells suppress this chromatin reader BRD4, which allows the cells to still arrest, but they can no longer upregulate the SASP, or the secretory program. And so here's a movie here. If you look at the proliferating cells, these red immune cells don't really recognize the GFP positive cells, but if they're senescent, you can see that they rapidly are killed. And that's SASP-dependent because if we or we suppress the SASP, we don't see this killing. So in this case, the immune surveillance component of senescence is leading to the elimination of senescent cells. And so again, many people have been interested in studying the role of senescence in tissues. I mean, we're learning a lot about where this process can contribute. So as again, if cells are damaged or acquire oncogenic events, types of injury, they can undergo senescence, leading to arrest in the SASP. And if that leads to immune surveillance, those senescent cells are cleared. And we think that's important for tumor suppression. And again, in wound healing or various types of regenerative programs. However, if the injury is sustained or the immune system doesn't clear the cells, senescent cells accumulate. And it's thought that these secreted factors cause tissue damage and tissue decline. And in fact, senescence in particularly older individuals has been linked to promoting cancer, diseases such as fibrosis, atherosclerosis, osteoarthritis, and certain neurodegenerative disease, and even organismal aging. And in fact, some exciting work now 5 to 8 years old from Jan van Derzen at the Mayo Clinic has shown that if one genetically eliminates senescent cells, these are in mice, one can ameliorate many of these pathologies. And so that's led to a whole interest in trying to develop strategies to do this in human patients. And so from a therapeutic pathway or modality or perspective, one might imagine that for cancer, one would like to induce senescence to try to stimulate resolution or tissue homeostasis, wherein some of these age-related disorders, one would want to eliminate that through, for example, a class of drugs known as senolytics. Now, I'm going to tell you 2 stories today, really one focusing on cancer and one focusing on other disease indications, where we've been attempting to try to modulate the senescence program. And the first study really is an effort that was stimulated by a whole group of postdocs in my lab together with a visiting scientist, Chuck Schur, that have really aimed to try to develop strategies to target, new strategies to target cancer, and inadvertently discovered one that could promote cellular senescence. And a lot of this work has been led by Marcus Ruschetti, but as I said, Joseph, Ricardo, Matt, Chuck, and John have played quite a large role. Now, the initial problem that we were interested in, this is really from thinking of Chuck Schur, was how to target Kras mutant cancer cells. And Chuck had an idea that it might be possible to develop a combinatorial strategy using 2 FDA-approved drugs. One that blocked the cyclin-dependent kinase inhibitor, CDK4/6, and another that acted upstream, downstream of activated RAS, but in the MAP kinase cascade, which should blunt the upregulation of the activator of CDK4/6, cyclin D. And the prediction is that would be particularly effective at limiting RB phosphorylation and lead to a very potent cell cycle arrest. And in fact, work from our lab showed that this was the case. This is just a proliferation assay of crystal violet, where one is looking at increasing concentrations of trametinib or palbociclib, and you can see this marked synergistic effect. Now, the surprise came when we looked at what was happening to the cells that were treated with the single agents or the combination I'll call through the rest of this talk TP. Not only did the cells arrest, but instead they also underwent senescence, here as measured by a marker senescence-associated beta-galactosidase. This senescence program was RB dependent, as we would have predicted, but could occur even in the absence of P53. And importantly, one also saw upregulation of this gene expression module for the SASP. You can see that really quite strikingly here. This is RNA-seq data, a heat map, where we've analyzed different cell lines that are Kras mutant treated with the vehicle, single agents, or the combination, looking at a senescence signature. And you can see that these combination-treated cells have a very different molecular state. They upregulate or downregulate genes associated with the cell cycle and upregulated those associated with SASP. And just as I showed you in the pre-malignant setting, if we take, in this case, lung tumor cells treated with the combination and do the co-culture assay I mentioned, you'll notice in the lower right-hand corner, the natural killer cells really attacking the senescent tumor cells, leading, as shown here on the left, to more cell death. This had a quite striking effect in vivo. So remember, senescence is a cytostatic program, but of course, if it triggers immune surveillance, tumors could regress. And we actually engineered mouse models that had a Kras mutation, no P53. They developed an aggressive lung adenocarcinoma, and then we could measure these tumors using micro-CT, where time 0, when we detected the tumor, and then 2 weeks later. And here's a waterfall plot showing that if we treat with the vehicle, the tumors grow as we expect. But each single agent has very little effect. Again, consistent with what we saw in culture. But remarkably, the combination has an effect, and it actually leads to tumor shrinkage. And that shrinkage is NK cell dependent, because if we deplete NK cells from the animals, now you see the drugs have very little effect. One of the secreted factors from the SASP is TNF alpha, and if we deplete that, we also see it disrupts the efficacy of our drug combination. So we see that this therapy that induces senescence can provoke an immune surveillance of tumor cells, and these data can explain how cytostatic drug combinations can promote tumor regression. Sorry for the typo there. Now, we went on to do these studies. That was in lung adenocarcinoma. In pancreas cancer, many of you know this is a disease that invariably is driven by Kras mutations. And we noticed, as we saw in the lung cancer setting, that these cells also underwent senescence. And you see that here in the treatment with the trametinib palbociclib combination. As we saw in vitro in the lung system, we could add NK cells to the tissue culture plate, and we could see them attack the NK cells. But in vivo, that didn't happen. NK cells did not accumulate in the tumor, and it had no effect, and depletion of NK cells had no effect on the drug combination. Okay, so that suggests there's something different between lung and pancreas. In fact, it wasn't the genetics, because the mouse models we used both had Kras activation and P53 mutations, suggesting there's something about the environment. Many of you may know that pancreas cancer is a very distinctive type of cancer in that it arises in the context of this dramatic desmoplastic reaction. And this has been considered to make pancreas ductal adenocarcinoma, or a PDAC, poorly vascularized, limiting access to the tumor by certain drugs. And it also creates an immunosuppressive tumor microenvironment. And so, again, it suggested that something was quite different in pancreas cancer compared to lung, and we wanted to understand what that was. So it's not that the drug combination didn't do anything. In fact, careful inspection of histological samples from either tumors treated with a vehicle or the combination, one could see here an increased number of red blood cells in tumors that were treated with the combination compared to controls. And in fact, if we stained these tumors with a marker, CD31, that's a marker of endothelial cells, you can see in the combination treated tumors that there are many more blood vessels. The lumen are more open, and these tumors are more vascular, and we could actually functionally show that. Now, we wondered whether or not this increased vascularization might be related to the fact that we triggered senescence in these tumor cells. And I mentioned that many of the factors associated with SASP are immune modulatory, but others of them, such as VEGF, PDGF alpha, or others, are also known to affect endothelial cell biology, either promoting proliferation or activation. And here what I'm showing you is a cytokine array where we're taking the vehicle, the single agents, or the combination, and you can see all of these factors showing increased secretion following the combination therapy, including, for example, VEGF. We wondered whether these secreted factors might stimulate the proliferation of endothelial cells in culture, so we could do a conditioned media experiment where we trigger the cells to undergo senescence, take media, add them to endothelial cells, and in fact, compared to serum depleted controls, you can see that they proliferate. And we also see increased sort of tube forming in a tube forming assay. Now, we wondered whether or not, in fact, this increased sort of this vascular remodeling we saw in the setting of combination therapy was SASP-dependent. And we took advantage of trick that I alluded to before, that the SASP gene expression module is NF-kappa B dependent, and we can knock that down using a short hairpin against the P65 subunit of NF-kappa B. And this is just a cytokine array showing you that in the combination that works. We reduce the secretion of many of these SASP-related factors. And if we look in vivo, these are mice that have PDAC, mouse models, where one can see in the controls this sort of vascular remodeling associated with the combination therapy. But if NF-kappa B is suppressed, that doesn't happen. You can see there's very few changes in the pore vascularization that we see in the controls. Again, that suggests that NF-kappa B is needed for the vascular remodeling and is consistent with the notion that the SASP factors that are NF-kappa B dependent are important for this process. So biologically, remember I said that PDAC is thought to be poor at taking up drugs because of this poor vascularization. And that's actually been shown to reduce the activity of a chemotherapeutic drug, gemcitabine, as shown initially by Ken Olive and Dave Tuvison. And so we wanted to ask whether or not treating with this senescence promoting therapy could increase drug uptake, presumably through this increased vascularization of the tumor. So with James Russell here at Sloan Kettering, we used C14 radiolabeled gemcitabine and triggered and treated mice that had tumors that were treated with the drug combination. Now, I'm just showing you vehicle and the combo here, and you'll notice by autoradiography really marked areas of greater drug uptake in tumors treated with the combination. And if we section through those, we see these regions of increased drug uptake are those that have more senescent cells and more blood vessels. And in fact, at different times, we see that this seems to sort of come from the outside and move inward over time. Nevertheless, that suggests we can get more drug into the tumor. Now, does that have any activity in terms of increased cytotoxic action? And so again, we looked at mice that have the KPC model. These have spontaneous KRAS-driven PDAC. And as I showed you for the lung model, if we just treat with a vehicle, these tumors grow over the course of 2 weeks. Gemcitabine by itself doesn't do much. In this model, really the combination doesn't do much either. There's no tumor regressions here. But if we layer in gemcitabine into the combination, now we see a marked anti-tumor response and some quite substantial tumor regressions. And in fact, a number of the mice live long. In fact, one was quite a long period of time. Now, we believe that the reason this drug works better when we've treated with the senescence combination is because of the new blood vessels. And to test this, we took advantage of an antibody, an antagonistic antibody against VEGF receptor 2 that at high doses blocks all new blood vessel formation. And when we add that on top, we see that now gemcitabine and the combination don't work any better, and there's no substantial survival advantage. Again, consistent with vascular remodeling playing an important role. Now, in addition to more blood vessels in these tumors treated with the drug combination, we also noticed that the endothelial cells had markers of activation, which often can occur during inflammatory response. One of those is p-selectin. You can see that in the upper right. Another one is VCAM1. And not only does this sort of suggest an altered state of the endothelial cells in these drug-treated tumors, but it's also known that activated endothelium can stimulate leukocyte uptake into the tissue. And indeed, when we look at T cells, in this case, CD3 positive T cells, in tumors treated with a combination, you can see substantial infiltration of T cells into the tumor. Now remember, at baseline, pancreatic ductal adenocarcinoma is a cold tumor. It is not inflamed, and T cells generally are excluded. Most of these cells were CD8 positive T cells. This is just quantifying them, shown here with the combination. Now, this increased T cell accumulation was also dependent on vascular remodeling, because if we have the VEGF2 receptor blockade, you can see that reduces the number of T cells that accumulate in tumors. Now, we did a lot of experiments I don't have time to go through, where we used, again, inhibitory antibodies to block different markers in the endothelium. Here's just an example of some of the results where we're looking at T cell uptake. This is just a positive result where we use antibodies that block VCAM1, and you can see that reduces T cells in the accumulation compared to what we see with the controls. Now, here we've just done an experiment to ask whether those T cells are actually doing anything. Now remember, this drug combination only produces a relatively modest survival advantage. And if we deplete CD8 positive T cells using, again, blocking antibodies, it has no effect on outcome. So even though the T cells are getting in, they're not doing anything. Now, why might this be? Well, we noticed that the T cells that were present expressed a number of exhaustion markers like PD-1 and CTLA-4. And so this would suggest that these T cells were exhausted and no longer able to see the tumor. Of course, that begs the possibility of testing checkpoint immune therapy that are checkpoint blockade, which are known to reactivate exhausted T cells, such as an anti-PD-1 antibody. And when we did this, we noticed that, in fact, the exhaustion markers went down. And consistent with that, the T cells that were present showed markers of activation. For example, in the lower right, expression of granzyme B. So what happens to the tumors? Well, here's the histology. Again, these are from KPC mice that have aggressive PDAC treated with the vehicle, the combination, or anti-PD-1 therapy. Now, it's known, again, in this model that immunotherapy alone doesn't do much. We don't see much activation here. But when we add that together with the combination of trametinib and palbociclib, you can see this marked cell death within these pancreas tumors, really something that we've never really seen before. We can monitor these tumors by ultrasound, and you can see this triple drug combination is starting to induce these pockets in the tumors that are consistent with necrosis. And that's blocked if we block with VEGF receptor 2 or an anti-VCAM1 antibody, or deplete CD8. So these drugs are no longer effective, all consistent with the possibility that by triggering senescence, we secrete factors that modulate the vasculature, leading to T cell uptake, which can be activated by checkpoint blockade. In vivo, this has a potent anti-cancer effect. So again, PD-1 alone doesn't do much. The combination doesn't do much. We start to see tumor regressions, quite substantial ones in some mice, when we now bring in checkpoint blockade. And in terms of survival, again, the controls don't do much, But now when we have the 3-drug combination, we're starting to get long-term survival in some of the mice, suggesting a potent anti-tumor effect. And again, that's blocked when we disrupt VEGF blockade. So to finish this part of the talk, what I've shown you is that senescence could be reestablished in Kras mutant cancer cells. And what the outcome of that is dependent on context. So in one setting, we can get NK cell attack, and other we can provoke T cell uptake. So distinct types of immune surveillance depending on context. But in pancreas cancer, this combination therapy induces vascular normalization, T cell penetration, and sensitization to checkpoint blockade. And it suggests that senescence induction can sensitize PDAC cells to new therapies, including conventional chemotherapy. Now, why might there be such dramatic effects when we trigger senescence in these tumor cells? And I just want to remind you that we believe that the native role of senescence is to participate in wound healing and wound repair processes, leading to tissue homeostasis. And we believe that by triggering this program in tumor cells, we can engage that sort of innate regenerative response, leading to tumor involution. Now, I'm just going to tell you about the second part of my talk, which again, so I told you here how we can have a drug combination that increases senescence in tumor cells, and that can lead to anti-cancer effects. But now I'm going to focus on the non-cancer indications, these tissue damage pathologies, such as fibrosis and atherosclerosis, where it's thought that senescence is a bad thing. And in fact, as I mentioned, genetic studies in mice have suggested if you eliminate senescent cells in this setting, it actually would help ameliorate these pathologies. Now, there's been a lot of effort to develop small molecule so-called senolytics to eliminate senescent cells from tissues. And this has shown promise. Of course, with all small molecules, specificity is an issue. And we wondered whether or not we could take advantage of the fact, as I already showed you, that the immune system can serve as a natural senolytic to eliminate senescent cells, to try to target these senescence-associated pathologies. Now, the way we decided to do this was really to take advantage of our collaborations with the Cellular Lab here at Sloan Kettering, who are experts in the development of Chimeric Antigen Receptor T cells, or CAR T cells, to target cancer. The work I'm going to tell you is really done by 2 trainees in my lab, Karina and Joseph, cancer biologists, together with Judith in Michelle's lab, who's an expert on CAR T cells. And the goal was to try to identify CAR T cells to eliminate senescent cells from tissues. Now, all of you that are sort of familiar with the cancer field know that CAR T cells have been an exciting new modality for the treatment of cancer. These, of course, are receptors that are engineered to target the molecule on the surface of a target cell. For example, CD19 in B cells to target B-cell acute lymphoblastic leukemia. These have a number of interesting features that, again, when expressed in T cells isolated from a patient, can engage those T cells to target the cancer, leading to its elimination. This can be curative in some patients. And so this process and these cells have been FDA approved, as shown here on the right, for a number of indications, and are widely used and being further developed to treat cancer. We were interested, as I said, in using them to target senescence. And the strategy that we chose was shown here on the schematic diagram, which was to identify cell surface markers specific to net senescent cells, to generate CAR T cells to target cells expressing those markers, and then to test whether they're efficacious. And also, of course, whether or not they would have any adverse toxicities. Now, we did a whole series of bioinformatics screening to try to identify candidate targets. We wanted to be high on senescent cells, not in vital organs. And we came across one shown here called the Urokinase plasminogen activator receptor, or UPAR. This molecule has been known for a long time. It's induced in a lot of damage settings. It's expressed on the cell surface through a GPI anchor. And when activated, or when bound by UPA, it stimulates activation of plasminogen into plasmin, which is important for the plasminogen activation system, which can eliminate blood clots and dissolve extracellular matrix in part of wound healing response. An interesting feature of this molecule is that a subset of it can be cleaved and secreted into the blood, so-called soluble UPAR, which can be detected in plasma. And in fact, has been used to mark sort of tissue damage in a kidney damage setting, clinical setting, which has actually subsequently been linked to senescence. In any case, we decided to try to test whether we could develop CAR T cells against UPAR and whether they would be senolytics. I should point out, we compared expression of the UPAR protein across a series of vital organs using the human proteome map. This is mass spec data that looks at a number of tissues. And we compared it to a number of proteins that are targets for other CAR T cells in clinical development. So I'm showing you UPAR here on top. You can see it's not widely expressed in most tissues. You'll notice quite marked expression on monocytes or macrophages. And we also know through immunohistochemistry there's weak expression on lung bronchial epithelial cells. But compared to, but similar to what is seen in other CAR T targets that are in clinical development. We wanted to confirm that, in fact, UPAR did go up during senescence and that it was expressed on the cell surface. And here I'm just using as sort of a control way to trigger senescence, the drug combination that I've spoken to you about before. The trametinib palbociclib combination triggers senescence. These are in the lung adenocarcinoma cells I talked about before. And if we do flow cytometry to look at cell surface UPAR expression, you can see massive upregulation of UPAR on these senescent cells. And we also note, using ELISA, the soluble UPAR also expressed in the media, raising the possibility, of course, that this could be a secreted biomarker of the amount of UPAR in an organism. We also confirmed that we upregulated UPAR in vivo. Again, this is using the lung adenocarcinoma model where we trigger senescence with the drug combination, and you can see UPAR is induced. And we looked at another series of settings in humans and in mice. Here's just some examples in mice where we've done oncogene-induced senescence. UPAR here is in red, or shown here in gray, where you can see, or sorry, green, you can see the upregulation of UPAR in these senescence indications. Here's a setting of liver fibrosis. This is one of the senescence-associated pathologies where senescent cells are thought to be deleterious, and you can see marked upregulation of UPAR compared to the normal liver. In humans, we've looked at a number of samples. Here's a setting of atherosclerosis from an atherosclerotic plaque, where you can see large amounts of UPAR expression. And here's human liver fibrosis, again, where one can see UPAR expression. And in fact, many of these other tissue damage pathologies have already been known that UPAR is upregulated. So here we have a marker, a cell surface protein that's upregulated in senescent cells. And so we set out to make CAR T cells. Here's a schematic of the CD19 CAR T cells that I'll be using as a control. And then engineered, different example that has the SCFV that targets UPAR, in this case, mouse UPAR. And we made chimeras to human and mouse CAR proteins. And this just shows you the mouse version here, that if we transduce this into T cells, we get expression. Here, the CAR is a MIC tag, which can be seen. And we're also developing, we also developed, as I mentioned, human CAR T cells that target mouse UPAR as well. This is just showing that we get specificity. This is an in vitro cell culture experiment where we've taken B-cell ALL cells. This is the NALM6 cell line that was used quite extensively to characterize the CD19 positive CAR T cells. And here, if we expressed it a murine UPAR, or plower is the gene name, cDNA, you can see that the UPAR CAR T cells kill these. The CD19 CAR T cells work as well, of course, because these cells express endogenous CD19, but untransduced T cells do not. The same BCL-AL cells that lack the murine UPAR cDNA still get targeted by CD19, but not the UPAR CAR-Ts. And here are cells, the KP lung adenocarcinoma cell lines treated with the TP drug combination that express endogenous UPAR. And you can see these also get targeted by the UPAR targeting CAR-T cells. Well, does this work in vivo? We did a couple of mouse models that I'll show you here. This is one that I would say is just a proof of principle. It's a setting we studied in a lab where we can induce senescence in the liver in a subset of hepatocytes of mice using a procedure known as high tail vein injection, high pressure tail vein injection. And we can deliver an activated oncogene, which stimulates oncogene-induced senescence over the course of a week or so. And when we did this, we wanted to see whether we could eliminate these senescent cells using the UPAR targeting CAR-T cells. We also, should say, delivered together with RAS-luciferase reporter, which allowed us to track ex vivo or externally the number of essentially senescent cells using bioluminescence. And you can see untransduced T cells don't reduce the bioluminescence burden, but those that targeting UPAR do. And you can see that on the right shown here, that we appear to be eliminating these senescent cells from the tissues. Histological analysis confirmed this. The mice treated with the untransduced T cells have senescent cells, are UPAR positive. They also express the NRAS oncogene, which was the trigger to drive senescence. But in the presence of the UPAR CAR-Ts, these cells are dramatically reduced. Using senescence-associated beta-galactosidase as a marker, we also saw a dramatic reduction in the presence of senescent cells. And we also noticed the accumulation of CAR-T cells in these regions where UPAR is expressed. Presumably, these are the regions of senescence, only with the UPAR targeting CAR-T cells and not the untransduced controls. You can see these green cells here. These are the CAR-T cells. And characterization of them by immunophenotyping showed that they have a high effector memory phenotype and low levels of exhaustion markers, consistent with them being active to target these UPAR positive cells. We did another model here, again, another proof of principle model, just where we can trigger senescence. This is the combination therapy model that I showed you before, where mice get lung cancer cells, then they get treated with a combination of trametinib and palbociclib that triggers a subset of the tumor cells to undergo senescence. And then we want to ask whether the CAR-T cells that target UPAR can selectively eliminate these cells from the animal. Now, if we just look at the presence of T cells in these lungs from these mice, we can see that when we have the UPAR CAR-T cells, there's an accumulation of CD4 positive, CD8 positive T cells. And consistent with them being active, we see an increased survival advantage of these mice in a Kaplan-Meier format when we treat with the UPAR CAR-T cells. Now, this isn't very substantial, but of course, we're not triggering senescence in all of these tumor cells. So really, to us, what was most important was whether or not we eliminated the senescent cells from the tumors. And I think you can see quite clearly here with also the CD19 CAR-T control that the UPAR CAR-T cells are very efficient at eliminating SA beta-gal positive cells from these tissues. Okay, so that says we can develop senolytic CAR-T cells. They work in vitro, they work in vivo. But would they have any activity in an indication, a model of an indication that we might think senescence is having a deleterious effect? Now, fibrosis is one of those settings, liver fibrosis being a main one. And there's a model of liver fibrosis that's been widely used where mice are treated with carbon tetrachloride. And over the course of 6 to 12 weeks, they develop fibrosis. With longer period of time, they develop hepatocellular carcinoma. And so we treated the mice with carbon tetrachloride, allowing them to get fibrosis. And then we treated those animals with the UPAR CAR targeting T cells or the controls. And when we look at the tissues from these animals, we first look at fibrosis, and you can measure that using a dye known as Sirius red. Mice that are given the untransduced T cells or the CD19 targeting T cells show fibrosis, as illustrated here. But that's reduced by 2 different doses of CAR T cells, almost completely eliminated shortly after treatment. We can also measure the SA-beta-gal positive cells in the controls, and that's eliminated following treatment with UPAR-CAR targeting T cells. We can quantify various features. The fibrosis goes down with these UPAR targeting CAR T cells. Senescence goes down. I showed you that on the previous slide. But we also see a reduction in soluble UPAR in the blood, again, consistent with this working. And we also see a reduction in liver damage as measured by the enzyme AST, which is very high in fibrosis and is reduced following treatment with the UPAR CAR Ts. So that not only says we eliminate fibrosis, it's consistent with reducing liver damage. Now, we did another experiment to characterize in more detail what was going on, where we incorporated, again, a luciferase reporter into the CAR T cells so we could track where they homed in the animal following injection. These, again, are all mice under the carbon tetrachloride fibrosis protocol. And if we just quantify the luciferase signal over time, you can see mice with the UPAR have a spike of signal around 5 days, and it's rapidly cleared. That never gets as high with the CD19 controls, which persist slightly longer. I think what's really striking is when we look at the animals, again, by bioluminescence. These are just different mice over time after CAR T injection. Again, all these mice have fibrosis. This is low and higher dose UPAR CAR T cells, untransduced, and those targeting CD19. Initially, many of these home to the lung, but you'll notice the UPAR CAR Ts then end up in the abdomen, and this is in fact where the liver is and where the fibrosis is before they're rapidly cleared. And you don't see that with the controls, again, suggesting that the CAR are going to the correct place. Now, of course, a critical thing with CAR T cells is they have to eliminate the target cell, the cells you want, but you don't want them to be toxic because, for example, there may be expression of the target on other normal cells that are needed for organismal health or viability. So we did quite a bit of analysis of toxicity, which I don't have time to go through in any detail. But we notice, as is seen in control CAR T cells, that mice with the low dose CAR T cells, this is 0.5 or 1 × 10⁶ CAR T cells, have some moderate infiltration of B cells and macrophages into the lungs. So you can see that a little bit shown here. But by and large are fine. They don't lose weight, and they seem to be histologically largely normal. We do notice, however, if we increase the dose substantially to 3 × 10⁶ CAR T cells, we do get appreciable toxicities. This can involve weight loss or temperature loss, and mice don't get a fever. They actually lose temperature, which is indicative of some form of toxicity. Histopathologically, there's a number of problems, again, primarily with the lungs. And these animals display features of cytokine release syndrome, which occurs actually in human patients that get CAR T cells as a consequence of rapid cytokine release, which causes fever and toxicity. Now, importantly, in patients, one can ameliorate these symptoms using the combinations of drugs that block cytokine release syndrome. And in fact, we see in our mice at the high dose treatment that these drugs can work as well. And here you can actually see this on the weight loss. You can see in substantial weight loss in mice with the UPAR CAR Ts, and again, that they recover more rapidly if we block cytokine release syndrome. So again, while high dose can be toxic, some of these toxicities can be mitigated with appropriate therapies. Now, I just want to finish by showing you one last indication, which is another liver fibrosis setting. And this is a setting that's associated with non-alcoholic steatohepatitis, or NASH, in human patients. This is a very serious disease that affects about 3 million people in the United States per year. And so this was a setting that we thought would be a very relevant setting to test whether or not these CAR T cells could be efficacious. We confirmed that, in fact, mice given a high-fat diet that causes NASH show accumulation of senescent cells. Many of them are UPAR positive, and we characterize these cells, and we previously shown that hepatic stellate cells are a prominent senescent cell type in fibrotic livers. But we also noticed in this model, much more than the CCL4 model, that there also were UPAR positive macrophages. Remember, this is the one normal cell type where we see UPAR substantially expressed. Now, we treated these mice again with high-fat diet to induce this NASH-related fibrosis, and then those mice got the CAR T cells, either the UPAR ones or controls. And as we saw in the chemically induced fibrosis model, we see that the senescence is, or the fibrosis is cleared. The senescence goes down. You can see that quantified shown here. And if we measure the function of liver by secretion of albumin, which is a good thing when the liver is working normally, we see that this CAR T therapy leads to increased albumin in the blood, suggesting it is having an effect on the integrity and function of the organ. So to finish, what I've shown you in this part of the talk is that UPAR is a cell surface protein. It's upregulated in senescence. CAR T cells against UPAR can selectively target senescent cells in vitro and in vivo, and they can have efficacy in certain senescence-associated indications. Of course, we're trying many more and are looking forward to that when our labs start up again. We're also intrigued by the possibility that soluble UPAR could serve as a secreted biomarker of this sort of CAR T response. I just want to end with the idea that the requirements for effective senolytic CAR T cells may be quite different than those that are needed for an potent anti-cancer therapy. Of course, for that, we want to get rid of every single cancer cells that need to be potent and persistent CAR T cells. But because senescent cells don't divide, and perhaps we'd be happy to eliminate even 80% of them, the CARs don't have to be quite as effective and they don't have to be as persistent. And we hope that in the long term might reduce their toxicities. So I'll finish this by returning to this summary slide where I've shown you a strategy in which we can increase senescence in tumor cells to facilitate immune cell attack, and a second where we can harness immune cells as a natural senolytic to selectively target senescent cells in vivo. We're excited, as I said, to pursue these. In the case of the therapy, we're interested in developing clinical trials. Of course, we have a long way to go on the senolytic CAR T cells, but we're also excited to pursue this as well. Now, I want to acknowledge, I mentioned the people who did the studies in the lab as I went along. I want to acknowledge my lab together with our colleagues. This is the way we have lab meeting now, as many of you out there do as well through Zoom. I'm showing you all their smiley faces the way I see them. And I also want to thank the people and funding agencies that have supported the work, HHMI, the NCI, Sloan Kettering, and the Lustgarten Foundation. So I'll stop there and turn it back to Donway and take any questions.
Thank you, Scott, for a very nice talk. We have a lot of questions coming in. Let me first start a question of my own. So we know senescent cells, they don't divide. So I wonder why, from the tumor's point of view, removing these non-dividing cells would be bad for the tumor itself.
So the question is, why would removing the cells from the tumor be bad for the tumor? So we think that, it's been widely debated as to whether cells can ever resume proliferation if they become senescent. And clearly getting rid of them, it would be a good thing to do if that were a possibility. It's also thought that, we do think that the tumor regressions is something that could be improved and enhanced with therapeutics. Also, we don't believe that all of the cells in the tumor undergo senescence, and we do believe that there may be some bystander effects also that would be eliminating the non-senescent cells in the tumors as well. And that's something we're actually studying in more detail now.
I see. Thank you. So the next question is, so in PDAC models where you also see metastatic cells, who's assuming they're metastasized to, for example, liver or breast, so the microenvironment would be different. So are the TP plus PDL therapy still effective in those cases?
Yeah, that's a great question. We're actually doing those experiments now. We've had mixed results. We do believe that once the cells leave the pancreas and go into the liver and the lung, that they now can be seen by NK cells. I mean, there we actually have completed a number of experiments where we know that, in fact, if, for example, the pancreas cells end up in the lung and TP therapy, NK cells will actually actively kill them as we speak. But we agree that it's certainly important to understand in more detail what's going on in metastases and particularly in the liver.
I see. So the next question is regarding the relationship with the NK cells in the co-culture. I think it also applies to in vivo. So the question is, does the SASP program also induce expression of NK receptor ligands on the tumor cells?
Yes, it does. It induces up a series of ligands and a series of cytokines that could modulate NK cell function. And we believe what is going on is in fact a combination of both, because we blunt some of the ligands and we blunt the cytokines. I showed TNF as just one example. You can reduce the response. So we think that, again, senescence, and David Rollet has actually shown this very nicely, upregulates the ligands, and that is important for the NK cells to recognize these senescent tumor cells.
And this listener is interested in the co-culture experiment. You showed very amazing effect for the NK cells. So this listener is wondering why. What about T cells, for example?
Yeah, so Ricardo in the lab has been working on that quite a bit, and we do see that there is we've put OVA in to get T cells that can specifically recognize an antigen in vitro. And when we do that, we see that the senescence environment does enhance T cell killing in vitro in a co-culture assay. It's interesting to think that might also increase, further increase the therapeutic index for the CAR T cells I mentioned in the second part of my talk. But yes, we do see that. We also note that the drug combination increases the expression of MHC class 1. And I realize there's some sort of, I guess, paradox in that we see in these cultures they can be targeted by NK cells. We see them be targeted by T cells, and there's sort of typically an inverse relationship between the expression of MHC class 1 and the ability to be targeted by NK cell attack. We don't know if the same cells are targeted, but nevertheless, those results are quite reproducible.
So this question is actually from a former CBG postdoc. So the question is whether or not you can go into maybe more details about how you select the cell surface markers for the CAR T strategy.
Yes. So the approach that we used initially was to take a series of RNA-seq data sets we had on different settings of senescence. So these were data sets we had in the lab for drug-induced senescence, oncogene-induced senescence, and so on, and then tried to look for transcripts that were upregulated in as many conditions as possible. And then we filtered those for those that predicted to be on the cell surface. And because lots of times cell surface proteins, their expression isn't always correlating with what's actually on the cell surface, we looked at this, we look at protein expression, and there were a few other ones that came through that sort of pipeline. But UPAR was the one that sort of stood out, particularly based on its lack of expression in vital organs. We do think there are other targets or proteins that could serve as targets. These are ones that we'd be quite interested in further exploring. But again, we think we have to, but anyway, that's how we got to UPAR.
I see. So actually, there's another question related to UPAR expression. So this listener is asking, UPAR apparently is also expressed in macrophages.
Yes.
So do you actually see the CAR T cell also clear tumor-associated macrophages?
So UPAR is expressed on a subset of macrophages, and we're characterizing in detail now. The UPAR positive macrophages that were present in the NASH model I showed do disappear. We don't know if it's because the CAR T cells eliminated them or because we eliminated other cells and they just no longer were present. And we're quite curious about the function of these cells and what they normally do. And I will say there is sort of an intriguing paradox in the senescence field that isn't understood. And that is many of the markers we attribute to senescence are also expressed in macrophages. And that's true for SA beta gal in a subset of macrophages. That's true for even P16, a cell cycle inhibitor that is broadly linked to senescence. And so there may be biology there, but we still don't know.
I see. So we actually have a lot of questions regarding UPAR and the CAR T cell therapy. So one of the questions is regarding other effects. So have you tried this therapy on aging associated disease models?
Well, we probably would have started that experiment. I mean, you know, that's an obvious thing to do. And of course, to do studies in aged mice takes time. But we're very curious about other indications, atherosclerosis, certain neurodegenerative conditions, and obviously aging as well. And those are experiments we'll do, but they're not done yet.
And related to that, maybe I guess this person is asking about potential side effects. So are those CAR T cell treated mice, do they have impaired wound healing or maybe increased blood clots?
Yeah, that's a great question. We haven't noticed the analysis of toxicity that we've done so far has sort of been in baseline mice that either primarily that have fibrosis. And we haven't noticed anything overt. I would imagine there might be some impairment or perturbation of normal wound healing, and perhaps other processes as well. And that's one reason why we're sort of intrigued by the possibility of having CAR T cells that don't persist very long. Again, in cancer, you really want them to keep going and target every last cancer cell. But here, it actually may be advantageous that they don't, because it would reduce the possibility of disrupting some of the physiologic settings where senescence plays a role.
So that's maybe a natural transition to the next question. So this listener is asking whether or not the CAR T cells express exhaustion markers. You know, depending on how long they are being exposed, they may also get exhausted.
So at the time point that I showed you, which I can't remember precisely when that was, I think it's about a week later, they did not. But I certainly wouldn't be surprised if they ultimately did. And I'd have to ask the people in the lab to confirm that. But we certainly detect them being activated in these sort of lesions of senescent cells, at least for a time.
I guess all these questions are sort of related. So this question is asking, so how long did you follow up on the liver fibrosis model, and whether there are trade-offs such as autoimmune disease?
So far, we've only done, I guess we did one study where we re-injected the CAR T cells, but we've gone out a couple of months in terms of the toxicities and looking at that. We haven't noticed any autoimmunity, but I'm not sure we necessarily would have seen that in the experiments that we performed.
I see. So this person, maybe coming back to the first part of the question, your talk. So this person is asking about BRD4. I think you mentioned in the very beginning that the SASP response requires BRD4. So this person is asking whether or not you've tried treating mice with the BET inhibitors.
So yeah, so BET inhibitors are molecules that disrupt BRD4, other BET proteins from chromatin. And that usually collapses the transcriptional modules that are observed in vitro. If we had a drug, for example, the famous JQ1, which is a BET inhibitor to senescent cells, the SASP module completely goes away. And in fact, we did an experiment in the oncogene-induced senescence model I alluded to, where we treated mice with JQ1, and the senescent cells weren't cleared. So in that setting, disruption of the bromodomain proteins did prevent the immune clearance of senescent cells. And again, it goes into this point that a lot of the molecules we study are context dependent. BRD4 protein or BET proteins can be very important, for example, in acute myeloid leukemia, to sustain the undifferentiated state. And there, if you inhibit them, they differentiate, and that's an anti-cancer effect. Here, we really see as the major, or a major thing these proteins do is to sustain this SASP program, and there the drugs do have an inhibitory effect.
Okay, so we have time for one last question. I'm saving the very difficult one for the last. So this question is, who is your favorite grad student?
The connection's going out. I don't know if I can answer that one. I'm going to divert that question to something that, since there were so many questions about CAR T cells, to remind people that in this series next week, Michel Sadelain, who's really one of the world's experts in this technology, will also be presenting. You can ask his questions, him some of those questions as well. And I'll have to think about the favorite graduate student question a little bit longer. But thank you very much for all those questions.
Yeah, thank you very much. And thanks to everyone who tuned in. That's all the time we have for today. And please join us again on Wednesday. Christine Meyer, our cancer biology program, cancer biologist, is going to give her presentation. See you, everyone.